Hey guys, today we're going to be talking about Chapter 8, Photosynthesis. Don't worry, we will go back and do Chapter 7, Cellular Respiration, after we finish this chapter. So I inserted some extra pictures for you guys to get an overall picture of photosynthesis. You know from freshman year that plants take water, sun, and CO2 to make glucose and oxygen. That's great, but now we're going to take a much deeper look at it. So I included a bunch of pictures for you to kind of get like a visual representation of what we're talking about. Because it's happening on a microscopic scale, it's very hard to be like, oh, here's physically what's happening. I can't like point to it and be like, well, that's photosynthesis. So I'm going to show you a whole bunch of pictures that I've inserted along the way. I think you should stop the recording, make sure that you go through them, and then it makes sense to you, maybe add them into your notes, because I really think a visual representation is going to be key to help you understand this process. Here's another picture that I found that I put in here for you. You've seen this generic little chloroplast picture probably before. I like the addition of the words on this. Again, I think that you should stop, add this to your notes, look at it, make sure that you understand what's happening from like our bird's eye view here before we get any deeper. You do have the light reactions and the dark reactions, also called the Calvin cycle, happening here. You remember those from the rap song that we had to memorize in class. But again, I'd stop this and make sure that you understand the basic idea of what's happening in our chloroplast before we go any further. Okay, so photosynthesis. It is a reaction that's going to convert light energy or solar energy into chemical energy, which in this case is going to be glucose. It's our food of plants. Okay, this is happening in our autotrophs. So autotrophs are things that can provide nourishment for themselves. So we are not autotrophs, we are heterotrophs, because you cannot stand outside and eat your lunch. You have to go physically to buy food, prepare food somewhere, and consume it. Okay, we're talking about plants and other photosynthetic organisms, some protists and some bacteria. Okay, so this process is highly organized, and it's highly organized because it's a whole bunch of these key players, and each one has a specific role, and you're going to see how complicated this gets later. But basically, we have a structural organization of our photosynthetic cells that includes enzymes and other molecules that are all grouped together within a membrane in order to help us carry out the process of photosynthesis. Okay, this organization allows for the chemical reactions of photosynthesis to proceed efficiently. Every single little moving part has its assigned job, and as long as it occurs correctly, then we're going to have glucose at the end of the day, which is what a plant's going to need. Okay, our chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria. This kind of goes back to the theory of endosymbiosis that we talked about when we talked about Chapter 4, the tour of a cell. So chloroplasts are the site of photosynthesis in plants. So we have this big overall reaction to make glucose. Now, where does it happen? It happens inside of the chloroplast. So the leaves are the major photosynthetic organ of our plants. If you think about on most plants, you know, the leaves are green. Well, green means that there's chlorophyll there. We're going to talk about chlorophyll in a little bit, but chlorophyll is what's actually going to be the pigment that captures this light. That's actually the next thing that this says here, right? So it's the green color from the chlorophyll. Leaves of our plants contain our chlorophyll because there's a lot of chloroplast there, okay? So the green pigment lies within the chloroplast. So wherever there's a chloroplast, it's full of chlorophyll. That's what makes a plant green. Okay, our chloroplasts are found mainly in the cells of our mesophyll tissue here in our leaves. So we have the interior tissue. Meso means middle. So it's the interior part of our leaves that have that mesophyll tissue, and that's where most of our photosynthetic reactions are going to be taking place. Okay, each cell of our mesophyll is going to contain roughly 30 to 40 different chloroplasts, and each one of these is carrying out the process of photosynthesis to provide food for the plant. So we're going to have carbon dioxide that enters and oxygen that exits the leaf through microscopic pores called stomata. Okay, you might have done a little lab where you did clear nail polish on the under part of a leaf, and if you peel it off, it looks like it has little tiny like bumps and holes in it. Well, that's because it does. Okay, just like you have pores in your skin and on your face, our leaves have these little tiny pores too. They're called stomata. They're just like your lungs, right? Your lungs are used for gas exchange. Well, plants don't have lungs, but they have stomata, and they're used for gas exchange. Notice I use the word exchange. It means that you're like getting and giving, okay? So while plants are bringing in carbon dioxide, they're releasing oxygen. Both of these gases are being exchanged by this particular structure, the stomata, okay? The chlorophyll is going to be inside of membranes of thylakoids that look like little tiny green stacks of pancakes that are inside of our chloroplasts. And the thylakoids can be stacked into columns. the stack I'm talking about looks like a little stack of tiny green pancakes, it's called a grana, or granum would be one. Okay, we also have chloroplast that contains stroma. It's the dense interior fluid. You can kind of think about stroma similar to like the cytoplasm of the cell. So like if you have the chloroplast, and then within that you have your thylakoids and your stacks of, thylakoids called grana. Around the outside of all that, you have like a jelly consistency of material called the stroma there. So it's kind of like the cytoplasm. It's kind of like a cushioning, but we also have reactions that occur in the stroma as well. So here's some pictures of what we were just talking about. We said that we have the photosynthetic organ of the plant as the leaves. And then specifically, we're talking about the mesophyll tissue, which is the middle layer, which you can see up here in this image. So it's the mesophyll layer that actually contains the chloroplast, roughly 30 to 40 chloroplasts within our cells here. You also see the little stomata. These are going to be used for gas exchange. Again, carbon dioxide will go in for photosynthetic use, and then as a byproduct, we'll have oxygen coming out. Here's the overall structure of our chloroplast. You can see that you have the little stacks of these little thylakoids here. Each little disc is a thylakoid. The whole thing is called a granum, or multiple is called grana. So inside of that, you can see that you have some open spaces. Okay, like the open space is going to be called our stroma, whereas we have the other material inside, like the thylakoid space and so on. Okay, so we're really going to be looking at tracking atoms and electrons as we go through the process of photosynthesis, okay? Now that we know where it's occurring, we're going to get into it a little bit more. And I don't want this slide to confuse you, because like even I read it, I was like, wait a minute, we're not going to talk about this. So let me just clarify for you. You know that plants take water, sun, and CO2 to make glucose and oxygen, okay? So you look at this equation here and you say, okay, carbon, awesome. Carbon dioxide, we got that going on. Also water, perfect. Okay, light energy called sunlight or solar energy, perfect. We're making some C6H12O6. It's a 1 to 2 to 1 ratio, carbohydrate called glucose. And then we also have oxygen, perfect. Okay, but what is this? Where did this come from? This is called water, H2O. Why is that a product of photosynthesis? If plants are walking around making oxygen and water, why do we need anything else? This is all that we need, plants. It's giving us everything that we need. Okay, so I don't want you to focus on the water, okay? Because do plants technically make a little tiny bit of water? Yes, it's water vapor though, so let's talk about it just briefly and then forget about it, okay? I want you to pretend like we have a big fat line here and then like this is gone. I don't want you to worry about this. Atomically, you need to understand that that's where the extra hydrogens and oxygen are going. It's water vapor, okay? But we're not focusing on this for the intents and purposes of the class. So essentially, you have oxygen that's released from the cleavage of CO2, okay? And that oxygen that's floating around can pair up with all these extra hydrogens that are also floating around in this whole reaction of photosynthesis. If you have hydrogen and you have oxygen come together, it makes water, specifically H2O, okay? So this would exit the plant as water vapor. It's a byproduct. A lot of textbooks don't even include it. They just leave it out altogether. This one happened to include it, and I don't want it to confuse you. Okay, so for the intents and purposes of this course, don't worry about it. You are worried about the process of photosynthesis as producing glucose and oxygen, and that's it. Okay, you do need to know where the oxygen comes from. Okay, understand that when you have the carbon dioxide and you cleave off the carbon here, you have a remainder of oxygen that can pair up with hydrogen and create this byproduct that we're never going to talk about again. Okay, but the oxygen that's actually released from the stomata, the oxygen that you and I are breathing in all the time, comes directly from the cleavage of water during photosystem 1. So water goes in and we're going to cleave it to create the oxygen that's being released. And we'll look at that later. You do need to know that, but you do not worry about this thing over here we're never going to speak of. Okay. So the splitting of water, like I just mentioned. So the chloroplast is really special because splitting of water is something that's a little difficult to do. So these chloroplasts are capable of carrying out this process in order to grab the hydrogens that they need, right? So chloroplasts are going to split H2O, called water, into hydrogen and oxygen. And they're going to incorporate the electrons from the hydrogen into sugar molecules called, glucose. And then they're going to be releasing the oxygen as a byproduct. So this is a very rare reaction that happens. But I mean, like, okay, plants take up most of our, planet, whatever, next to water. So like, is it really that rare? But like, other creatures can't do this. So this is pretty cool that plants can do this, right? So they're essentially going to just hang on to those hydrogens in order to use them as a source of electrons to help create their sugars. But then that oxygen they don't really need, so they get rid of it like almost right away, right? So it's very early on in the process. And we benefit from that because yellow, we need to breathe in that oxygen, okay? So here's the overall process of where everything is coming from. So like I said, you need to know that we are going to take water and we're going to break water. And what are we going to do with that? We're going to take the hydrogens and we're going to utilize them for our sugar and then this other thing we're never speaking of. And we are also going to use that oxygen as a byproduct to release to the atmosphere for our, other organisms that need to live on oxygen. Okay, so this is where our reactants are getting broken down and where they're donating all of their atoms. Okay, so photosynthesis is a redox process or a redox reaction. So if you've taken chemistry, you know that this is a reduction and oxidation process. So photosynthesis reverses the direction of electron flow compared to respiration. So basically it's just an opposite happening here. So photosynthesis is a redox process in which water is oxidized and carbon dioxide is reduced. And we're going to look at a picture of that in just a second, and it's going to make more sense. Okay, so photosynthesis is an endergonic process, which means we need the input of heat or energy for it to happen. The energy boost is provided by light, okay, the photon of light that's going to go in and excite our electrons. So here's what a redox reaction looks like. So this happens to be photosynthesis, and notice that there is no naughty no happening on the end of this here. This is the correct photosynthetic equation, okay? So we have carbon dioxide that's becoming reduced. What does that mean? There's an acronym for this. It's oil rig. If we're talking about reduced, that's the rig part, R-I-G. Reduction is gain. What are we gaining? We're gaining electrons in the form of hydrogen. So we have carbon and we have oxygen, but now all of a sudden we have hydrogen, because we're gaining electrons, okay? Reduction is gain. Reduction, we're gaining electrons in the form of hydrogen here. Oxidation, the oil part of oil rig, O-I-L, oxidation is loss. So here you have hydrogens and you have oxygen. And now at the end, you only have oxygen. It's oxidized because oxidation is rude. Oxidation is loss, so you were losing electrons in the form of hydrogen here. So just say that a few times to yourself. Oxidation is loss of what? Electrons. Okay, so electrons, literally so rude. I'm just going to keep going because it's going to take me forever. Oxidation is loss of electrons in the form of hydrogen. We're losing hydrogen here. Perfect, that's oxidation. Here we have reduction because reduction is gain of electrons. We are gaining electrons. This is so rude. I'm so sorry. So we're doing that in the form, again, of hydrogen. So carbon dioxide, now we're gaining hydrogens here. It's being reduced to become sugar. Okay, so our two stages of photosynthesis. This is a entrance of the crackheads. Echo and Reagan, my monsters. A two-stage process here. This is a preview. This is a bird's eye view. So we're starting like big screen and we're going to go zoom all the way in and focus on just one reaction at a time in just a second. But the overall preview. So photosynthesis consists of our light reactions, the photo part of photosynthesis, using light to make something. Okay, and then the Calvin cycle is the synthesis part where we're actually going to be creating, combining carbon with other molecules in order to create our G3Ps, which will eventually turn into glucose. Okay, so we have the light reactions in the thylakoids. That's going to be where we have the splitting of water, which again is really rare and really cool, and then we're going to be releasing oxygen. Okay, that's the first step. The light reactions are the very first step. Think about what I just said, though. We're releasing oxygen in the first step. If there's two steps in the first step, what if you made glucose in the first step? Why would you need a second step? If the plant got what it wants, why the heck would you go on and do another whole step? You wouldn't. So there's two reactions that happen. And the first reaction, you get a waste product. That's why you have to go on and do the second step. And that'll make sense as we look at a picture, and I'll point it out to you there as well. Okay. So again, in the light reactions, we have the splitting of water and we have the release of oxygen. Very important. Okay, we are going to reduce the electron acceptor, NADP+, to NADPH. Now again, what is reduction, reduce. What is that? RIG, reduction is gain. Gain of what? Electrons. How do we gain an electron? What does it look like? It looks like an H called hydrogen. So now we have NADPH because we just got reduced. We're adding an H because reduction is gain, rig, oil rig. Okay, so generate ATP from ADP by adding a phosphate. So this is photophosphorylation. Okay, so we have ADP floating around. We're adding another phosphate group to go through photophosphorylation because we're utilizing light in order to generate ATP. Photo is the light part. Phosphorylation means to add a phosphate group. We're adding a phosphate group to ADP to create ATP. Okay, so in the Calvin cycle, that's the second step. They're also called the dark reactions, which is a little bit misleading, but light only interacts with the light reactions, okay? Light is not utilized in the Calvin cycle, okay? So the Calvin cycle that happens in the stroma, remember that that's like equivalent to the cytoplasm, okay, forms sugar from carbon dioxide using that ATP that we just made in the light reactions and that NADPH that we just made in the light reactions. So if we didn't do the light reactions, we wouldn't have these two things, which means that we would not make sugar. Okay, so we're going to form sugar from carbon dioxide, which is in the air, okay, and that enters through the stomata using ATP and NADPH. And where did these two things come from? ATP and NADPH, they came from the light reactions, okay? So the Calvin cycle begins first, so there's 3 different phases. The first phase is called carbon fixation, and it's going to incorporate carbon dioxide into organic molecules. Okay, here's an overall reaction I was talking about. We have like light reaction here, and our Calvin cycle or dark reaction. We're going to call this step 1, and we're going to call this step 2. So in step 1, if we made glucose here, why the heck would we do all of this? What is this garbage? If we made glucose right away, that wouldn't make any sense, right? I'm going to tell you that this will be a quiz question where all of these will be like little A, B, C, D, and it's going to be like, oh, which one of these represents oxygen? Oxygen is a product. You know that. So it's going to be coming out. So you got a 50-50 shot. It's either the green arrow or the blue arrow. But think about it. Does the plant want oxygen? No. So it's going to make it first. Because if it made glucose first, why the heck would we do all of this jazz? We would not want to do that. It's just a waste of energy. Look at this. We're using ATP. Oh my gosh, that's a waste of literal energy. Okay. So again, if this was like all blanks, it would make sense that you're making the waste product first. Because otherwise, why would you do a second step? If you made glucose here, it would make no sense to keep on going, okay? So just remember that. Also, Calvin cycle, CC, C goes into CC. It's very simple, okay? Also, you know, the light reaction's really, really cool because they can split apart water. It's really neat, and that's where our oxygen comes from. Okay, so the light reactions convert solar energy to the chemical energy of ATP and NADPH. Why do we need those two things? Because that's how we're going to create sugar in the Calvin cycle, okay? So chloroplasts are solar-powered chemical factories, okay? Their thylakoids transform light energy into the chemical energy of ATP and NADPH. And you remember this from our little rap song as well. It's so nice that I made you learn that. So here's another picture that I put in, okay? We are looking to track our NADPH and our ATP. This is the overall process of the light reactions. This is a little bit more complicated than this picture. This is, oh, light reactions, cute. It's 2 words and it's on something green. Okay, but like this is what's actually going on there that we're going to talk about, okay? So like I said, what are we doing here? It's a chemical factory to make NADPH and ATP. And where are those 2 things going to go? They're going to continue on and they're going to go into the Calvin cycle, step 2, the dark reactions, okay? That's exactly what's happening here. We're looking to create these 2 things to then push them into the Calvin cycle, okay? So this is actually what's happening. This is another picture that I snatched from somewhere else online. The description's really great. The picture's really great. Again, please pause and look at these, especially after I already talk about it and you can go back and be like, wait, what the heck did she say about this? Oh yeah, that makes a little bit of sense. I'm gonna stare at this picture and hope that through some sort of diffusion osmosis sort of electrical impulse, something that it gets into my brain hole. So please make sure that you look at these, okay? All right, there's a bunch of slides in here that I took out about the visible spectrum of light and spectrophotometers and blah, blah, blah. I don't care about that. I'm going to refer to the photosystems as P680 and P700, and I just want to tell you what that means. 680 and 700 are referring to nanometers. That's what this little NM means. If we're talking about the visible light spectrum, like this is where like 680 would be like, like yay, and then like 700 is like, okay, cool. So if it's here, we're actually going to be reflecting green because it's mostly like absorbing red mostly, okay, but then that's why we see green. Okay, let's talk about that. So that's the visible spectrum of light, just so you know. Cool. Okay, P680 and P700 is literally talking about the photosystems and then the nanometers of light that they absorb. Okay, cool. So the light receptors here. Pigments are substances that absorb visible light. Guess what? Visible light, boom, there it is. Okay, the rainbow, gorgeous, Roy G. Biv and all that, you know that. Okay, great. Okay, so different pigments absorb different wavelengths. Okay, a pigment that we're focusing on, chlorophyll, because why? It's in plants. What are we talking about? Plants, because we're talking about photosynthesis. Okay, so wavelengths that are not absorbed are reflected. So if we're over here, we're absorbing these, we're going to reflect things like this. This is why you get, the colors like this direction. Okay, no, you don't have like purple leaves. Well, sometimes those are actually really cool looking. Mostly green. We're focusing on like green leaves because they do not absorb green light. They reflect it. Okay, so leaves appear green because chlorophyll reflects and transmits green light. Focusing on the green because chlorophyll is what? It's green. Okay, it's a pigment that absorbs light, but not green light because it reflects it, because that's what you see. So that's what's happening here. That's why we see green. Beautiful, love it. Okay, so absorption spectrum, blah, blah, blah. I'm not going to be talking about all that, but we do have chlorophyll A and we do have chlorophyll B and these things called carotenoids. Okay, so chlorophyll A and B absorb very similar wavelengths. Chlorophyll A is our main photosynthetic pigment. Chlorophyll B is a secondary pigment, and carotenoids are kind of there for like the aftermath, okay, to help prevent light damage and things to our plants, but we'll talk about those more in person later. You need to know chlorophyll A is the main photosynthetic pigment in plants. Okay, hey, look at that. I just said that. It's like I read this before. Okay, so chlorophyll A is the main photosynthetic pigment in plants. Beautiful. So accessory pigments like chlorophyll B, like I said, we don't really use that a whole lot, but it does broaden the spectrum of light available for photosynthesis. And then there's a little bit of a structural difference between chlorophyll A and B. It's on the next slide. It's literally like 2 atoms difference. Okay, and then our carotenoids absorb excessive light that would damage the chlorophyll otherwise. And it's also the reason why we can get different variations in color in our plants as well. Okay, so this is what I was talking about, the structure of chlorophyll A and chlorophyll B. So like the only thing that's different is this little group right here. Instead of a methyl group, it's like this little like CHO group. Okay, beautiful, great. Excitation of chlorophyll by light. This is where it starts. It's happening. It's about to happen. Okay, so when pigments absorb light, pigments called what? Called chlorophyll, mostly chlorophyll A. Okay, it absorbs light. That's what it does. Okay, it goes from a ground state to an excited state, which is very unstable. Okay, so when excited electrons are going to fall back down to their ground, not cray, but just ground state, okay, protons are given off. And it's an afterglow called fluorescence. And we'll look at a picture of that in a second. Okay, so if illuminated, an isolated solution of chlorophyll will fluoresce or give off light and heat. Boom, look at that, fluorescence. Okay, we don't see plants that glow because there's somewhere for all of that energy to go, and it's called the electron transport chain, and we're gonna look at that in a minute, okay? But we don't see glow-in-the-dark plants all the time because there's somewhere for that energy to go. Now, if it was just in a contained flask here, like this Erlenmeyer flask that's full of a fluorescent material, it would fluoresce, it would glow because it has been excited and it's illuminated. Okay, so photosystem, we're going to talk about 2 of them. Like I said, P680 and P700. P stands for photosystem. So we're going to be talking about 2 different photosystems. So what the heck is a photosystem? It is a reaction center. It's a complex of a whole bunch of different moving parts and enzymes, and it's associated with harvesting light complexes or light harvesting complexes. So a photosystem is a reaction center. That's the P680 and the P700 parts. Okay, it's a protein complex that's surrounded by light harvesting complexes. Well, this makes sense because, plants do photosynthesis. It's when we harness light and then make sugar. So, like, we need light harvesting complexes Beautiful. Okay, so these are pigments called what? Called chlorophyll, mostly chlorophyll A, that are going to transfer energy of the photons to the reaction center that's called P680 and P700. Okay. So this is kind of like what's going on here. This is a little complicated, like the molecular structure of this, like the actual beta sheets and alpha helices and things. Okay, cool. We're going to look at this picture because it's a little bit more friendly. We have a photon of light coming in. It's always like this little lightning bolt. Is it actually lightning? It's just sunlight. It's visible light. It's sunlight, okay? Here we have our photosystem, okay? So we have the purple part here that has our light harvesting complexes, pigments, things like that. And then in the center, you have your reaction center complex. Okay, so that's the overall structure of our photosystem. Okay, a primary electron acceptor in the reaction center accepts excited electrons and is reduced. Again, reduction is gain. RIG, oil rig, RIG, reduction is gain as a result. Okay, solar powered transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reaction. One more time for those of you in the back. We have light, it's coming in. It's going to go into chlorophyll A. That chlorophyll A molecule is going to hand off the excited electrons to a primary electron acceptor. Primary means like first electron acceptor. Makes sense because it's accepting the electrons from who? From the chlorophyll A. Okay, that's the first step of the light reaction. Okay, there are two types of photosystems in the thylakoid membrane, as promised. Photosystem 2 functions first. Now, before you're like, wait, 2 comes number 1? Yes, because they were named in the order that they were discovered. Okay, so photosystem 2 was discovered after photosystem 1 had already been named. Why didn't they change the name? I don't know. I wasn't alive. My parents probably weren't even alive. Okay, so photosystem 2 actually comes first. Just remember that. It's not that complicated. Okay, 2 comes 1. Okay, photosystem 2 comes first. And it's best at absorbing a wavelength of 680 nanometers. As promised, that's where that P680 comes in. P680 is the reaction center of photosystem 2 because it's absorbing that wavelength of light, okay? Alternatively, photosystem 1, which actually comes second, 2, and then 1, is called P700 because why? It absorbs wavelength 700 nanometer light. Okay, beautiful. That's the reaction center there. Okay, so we have this linear flow of electrons, and we're going to be following this in order to create our ATP and NADPH because those are very important. We're going to pass them off into the Calvin cycle. Okay, linear electron flow involves the flow of electrons through both photosystems, 2 and then 1, because reasons, to produce ATP and NADPH using light energy or solar energy. Okay, so here's the steps. Linear electron flow can be broken down into a series of steps. There's something like 8 of them. So here we go. Okay, a photon hits a pigment. Light hits chlorophyll A. And its energy is passed among pigment molecules, other chlorophylls, until it excites P680, which is the reaction center in photosystem 2, which comes first. Okay, an excited electron from P680 within photosystem 2 is transferred to the primary electron acceptor, the first thing that actually holds on to those electrons, and we now call it P680 plus. Don't get too bogged down by that. I'm not too worried about that last part. Okay, step 3. Water is split by enzymes, and the electrons are transferred from the hydrogen atoms to the P680 plus, thus reducing it to just P680. As a result, we have oxygen that is released as a byproduct. So again, where did that oxygen come from? I just told you because I'm dumb. It came from water, okay? So oxygen is released as a byproduct. Why? Because we split oxygen. Nope. Words are difficult. Because we split water, we use the hydrogen to make this plus sign go away by reducing it and adding in electrons, because electrons are minus, so we have a plus plus and a minus minus, and it's going to get back to 0, P680. And then after we remove these hydrogens, we have these left over in oxygen. Okay, oxygen is released as a byproduct, and it's in the first step because the plant does not want oxygen. Okay. So this is where we're at, right? We got the light coming in, it hits photosystem 2, the chlorophyll is going to have the excited electrons, we're going to start passing down the electrons, we have the splitting of water as it's coming in, and we're releasing oxygen. Beautiful. Okay, here's another example of that, because this is what you're used to, but this is what we just said, okay? You got light, it's coming in, all right? You got all of the chlorophyll molecules that are bouncing around. All the electrons are getting all excited and they're passing them down. And then we have the reaction center, P680, is going to accept those electrons. Okay. And then we're going to have the splitting of water and the release of oxygen. And then look at what we have to go through next. Wow. It's a lot. Okay. So each electron falls down an electron transport chain from the primary electron acceptor of photosystem 2 all the way down to photosystem 1. So essentially, it's just like passing a ball between a whole bunch of different people, and the ball is an electron. Okay, the electrons are very excited. It's like passing this energy down of these excited electrons down a huge conveyor belt of different enzymes and protein complexes in order to eventually reach photosystem 1, okay? Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. Diffusion of these H+ hydrogen protons across the membrane drives ATP synthase. Okay, this is when we're talking about electrochemical gradients, back when we talked about Torricelli in chapter 4, when we hit cellular transport. Okay, if we're gonna pump a whole heck of a lot of these protons into the thylakoid membrane, and as they come back, because they're going to want to drift back from high to low concentration, because now we've created a very high concentration, diffusion will drive them back down the concentration gradient. They're going to spin this rotating complex called ATP synthase that's going to create ATP. We'll look at that later. Step 6 in photosystem 1, which is the second photosystem, because reasons. In photosystem 1, okay, like photosystem 2, the transferred light energy excites the reaction center called P700. Again, the number is the wavelength of light that it absorbs, causing it to lose an electron to an electron acceptor. So now it's the same thing, P700 plus. Okay, P700 plus accepts an electron passed down from the photosystem 2, which actually comes first, via the electron transport chain, which will return it to just regular P700. Okay, so the excited electrons fall down an electron transport chain because it's transporting electrons in a chain of events from the primary electron acceptor of photosystem 1 to the protein ferredoxin, which is abbreviated as FD, ferredoxin. Okay, so that is at the very end. That is the primary electron acceptor at the very end. Okay, so we have our electrons that are transferred to NADP+, reducing it, RIG, reduction is gain. We're adding a hydrogen, which is really an electron. Okay, reducing it to NADPH, and it's available for the reactions of the Calvin cycle, because we're going to send NADPH and ATP into the Calvin cycle to help us turn that carbon dioxide into glucose, or G3P first, but then glucose. Okay, this process also removes an H+ from the stroma, which again is like the cytoplasm. Okay, the energy changes of electrons during the linear flow can be represented in a mechanical analogy. I took that picture out. You just need to know that these excited electrons are consistently passed from one acceptor to the next until it reaches the end of the electron transport chain, because it's back into its like ground state. That's it. We're slowly decreasing in energy as it's, quote, falling along the whole way. Okay, so we're going to take a time out for just a second to compare the chemiosmosis in chloroplasts and mitochondria. Okay, what the heck is chemiosmosis? Okay, remember osmosis? It's a thing like water moves from high to low concentration. Chemiosmosis is basically when we have chemicals moving from high to low concentration and it's used to generate energy for something else. So in chloroplasts and mitochondria, we're going to generate ATP by chemiosmosis, but they use different sources for this energy. So in the mitochondria, which is not what we're talking about right now, but we're going to talk about it when we talk about cellular respiration, because that's where ATP is created, powerhouse of the cell. Okay, the mitochondria is going to transfer chemical energy from food to ATP because we eat things and then make ATP. Actually, this also occurs after we already generate that glucose. That is a food source that we're going to turn into ATP. Okay, chloroplasts transform light energy into the chemical energy of ATP. So we go from food to ATP, and then chloroplast is light to ATP. So like, yeah, there's a pretty big difference, okay? But the spatial organization of chemiosmosis differs between the chloroplast and mitochondria, but they also show some similarities. So while the overall mechanism is a little bit different, there are a lot of similarities. And when we actually learn that process, you'll see that they are kind of similar, and you'll be like, what the heck? These are too close. I'm going to get them confused. You're right. So make a really good study guide card for the test. Okay, pictures are very helpful. The pictures worth 1,000 words. You're right, they sure are. Okay. Okay, so in the mitochondria, we have protons that are pumped to the inner membrane space and drive ATP synthesis as they diffuse back down into the mitochondrial matrix. And in the chloroplast, we have proteins that are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. Guess what? That sounds real similar. It's because they are. Okay, so the process is different because we're going from food energy to ATP and then light energy to ATP in the chloroplast. So the mechanism's a little bit different, but like how we actually go about creating this ATP is pretty similar. Basically, it's an electrochemical gradient that's going to diffuse back down through the concentration gradient in order to generate ATP. At the end of the day, that's the moral of the story. Again, if this sounds like I'm just spitting chemistry, I am, but also pictures are helpful. So go back and look at the pictures, look at your textbook, read it. It's really helpful. It's a whole bunch of words that like really make it clear. Okay, so we have ATP and NADPH that we produced in our light reactions. Okay, they're produced on the side facing the stroma. Okay, and then that's where the Calvin cycle takes place. So they're produced on the correct side of the membrane, so then we can go ahead and just like feed them straight on into the Calvin cycle, which is really nice. Okay, so in summary, the light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. Okay, remember that we split that water. We took those hydrogens in the form of, like electrons, okay, and then we slapped them onto NADP plus to create NADPH. And what do we do with that oxygen? We released it through the stomata for us to breathe, okay? So again, overall process, we just talked about this. And that was a lot of words and a lot of carbon dioxide that I just created in order to say those words to just talk about this. But why is this important? Because of this. This ATP and this NADPH, we just had to create that from here in order to drive this next reaction, okay? So we're going to use these products to go into the Calvin cycle because the Calvin cycle is even worse than the light reactions, okay? That's where we're actually going to create the sugar for the plant, and that is what the plant is after, and it is very important. Here's the picture straight out of your textbook. Okay, this is actually a really great image of everything that's going on here. There's even more words associated with this than what I said. Okay, we got the photosystem 2 happening here. You got your photon of light going in. It's exciting the electrons in the chlorophyll. You got your reaction center. You got your splitting of the water to release the oxygen. Okay, it talked about bringing more of our, words are difficult, protons into the thylakoid space. You have these other like handoffs that are happening here, like plastoquinone and cytochrome C complex, and then plastocyanin, I believe is what that one's called. And that's going to hand it off then to photosystem 1, which the terminal electron acceptor is going to be our ferredoxin, which we did talk about, which is abbreviated FD. Okay, and then we have our NADP plus reductase. Reduction is gain. Okay, we're going to add on, we're gaining an electron essentially here, okay? So we're creating NADPH, and then again, we're creating this like increase of our hydrogen here, which is going to flow back out because it's a high concentration to a low concentration. As it goes back out through this transport protein, it's going to create ATP. Okay, ATP synthase means to create, it's the enzyme that creates ATP by photophosphorylation, and that's where we create our ATP. Guess what? These two products are now going to move into the Calvin cycle. Again, pictures are worth 1,000 words. Please make sure that you pause and look at the pictures. They're really important. Okay, this is an overall picture of the Calvin cycle, and I really like this one because it includes a little 1, here's what's happening. 2, this is what's happening here. Okay, all of this is really important, and I like that the pictures are all color-coded. I like that there's steps for you to read along the way, which are like just a little bit different than what it says in your textbook. Just make sure that you go back and look at these, because we just talked about the light reactions. Now the Calvin cycle is something completely different, and you're looking at that and you're like, okay, cool, it's a circle. You need to come back to this after I'm done spitting some game about all of the Calvin cycle steps, okay? Here's another picture for you, a little bit simpler. You can see, oh, great, 1, 2, 3, 4, 4 steps. I got this, okay, but like it's a lot still. You know what I mean? Okay, so you make sure that you come back and that you understand what's happening. Yes, you have your, overall phases that are happening here, but like this one does it in 6 steps. Like, do I care about the number of steps? Not exactly, but I want you to know what's happening along the way for the entire process. Okay. This is also a little bit janky, but I really like this picture because it's basically broken down into 3 steps. So you have carbon fixation, reduction in sugar production, and you have regeneration. Those are the 3 main steps of this overall process. And like here, it's written out very well for you. I would pause this, zoom in on it, flip it if you're on a smartphone or a tablet or something, and you can like, zoom in on it. I recommend that. Add anything to your notes that you don't already have there, and then, come back to this when I'm done creating some carbon dioxide about this whole process, okay? All right, so we previously talked about the light reactions. We can do it. This is the second half. We're gonna make it, okay? This is the Calvin cycle, also called the dark reactions. Okay, the Calvin cycle uses the chemical energy of ATP and NADPH that we just created to reduce carbon dioxide. Now, what is reduction? Reduction is gain. What are we gaining? Electrons. What are we gonna become? C6H12O6, called glucose. Okay, so the Calvin cycle, like the citric acid cycle, don't worry about that. Just ignore that. We'll come back to that later, another time. The Calvin cycle regenerates its starting material after molecules enter and leave the cycle. This is important, but we're going to look at some pictures and you'll see why it's important, okay? So, blah, 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 citric acid, who cares? The Calvin cycle is anabolic. What is anabolic? Think about anabolic steroids. I like to pick things up and put things down. It's how you grow muscles, anabolic steroids, like, like illegally, cool. Okay, so anabolic means building reactions. Why are we building? Because we have CO2, we're going to turn it into C6H12O6. That's definitely a larger molecule. We are building that up, okay? It builds, anabolic builds anabolic. Oh my gosh, it's like naming the same thing, because they do. Okay, it builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. Okay, so carbon enters the cycle as carbon dioxide. Where did this come from? It came from the atmosphere, from like our little hot breath when we're breathing, and I'm speaking right now and I'm generating a lot of it for the plants. Save the plants, okay? So carbon dioxide enters the leaves through the stomata, okay? So carbon enters the cycle as carbon dioxide and leaves as a sugar named glyceraldehyde 3-phosphate, okay? For net synthesis of 1G3P, which you need 2 of those to make a glucose, okay? So for the net synthesis of 1G3P, okay, the cycle must take place 3 times, fixing 3 molecules of CO2. Now, if you're intelligent here and you can say, well, if we need 2 G3Ps and you have to use 3 molecules of CO2 to make 1, then I need 6 molecules of CO2 to make 2 G3P, also 6 CO2, 6 C, okay, C6H12O6, okay, because we need 6 carbons. I hope you got that. So the Calvin cycle has 3 main phases. It has the carbon fixation stage, the reduction stage, and the regeneration of the CO2 acceptor stage. 3 stages, okay? Each textbook calls them something like a little bit different, which is why that weird janky sideways picture calls them different steps, but what's happening is still the same. Cool, okay. Phase 1, carbon fixation. You know this again from our rap song. Again, you're welcome, okay? So carbon fixation involves the incorporation of carbon dioxide molecules into ribulose bisphosphate. This is why we use acronyms, because no one wants to say that. Okay, ribulose 6 bisphosphate. This means like two phosphates. Okay, so this is RUBP. 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 Using the enzyme Rubisco, which we learned in our rap song. Okay, it's like the most common enzyme like in the world because plants. Okay, so RUBP is ribulose bisphosphate. So it involves the incorporation of carbon dioxide molecules into ribulose bisphosphate using Rubisco. So we're going to turn CO2 into this using this. Great. This picture is not really helpful here. Thanks, textbook. All right, here's the overall thing. We're going to have 2 turns of this overall, essentially. So we're inputting 3 carbons. This is this little circle, spoiler alert, it's a carbon. We're gonna input 3 carbons as 3 CO2s, 3 carbon dioxides. Now remember, that's gonna make 1 G3P, which I said earlier. We need 2 G3P. So this process, it gonna happen twice, okay? So input 3 carbons as 3 carbon dioxides. You see Rubisco, it's an enzyme. So we have RUBP that's going to meet up with our CO2 in order to create, see we have 1, 2, 3, 4, 5 carbons in RUBP, ribulose bisphosphate, okay? That's what this is. Bisphosphate means 2 phosphates. Oh my God, look at that. It's like the name matches. Okay, CO2 has 1 carbon. So we have our 1, 2, 3, 4, 5. Oh my God, 6, because it came from right here. Okay, we're just showing it for 1, but this is really happening 3 times at each of these steps. Okay, and then we're going to have 3 phosphoglycerate. Okay, this is a 3 carbon. Basically, you're like cleaving this. Okay, and then you're going to add an ATP in here, and you're going to slap one of the 3rd phosphate onto a 1,3 bisphosphoglycerate, because here phosphoglycerate means that there is 1 phosphate at the end of a glycerate molecule here. So essentially we just like cut this in half essentially, okay? So then here 1,3 bisphospho means in position 1 and position 3 we have 2 phosphates. Position 1, position 3, oh my God, it's like it matches. Okay, we're going to go through, we're going to utilize that NADPH that we brought in from the light reactions. We're going to oxidize this in order to oxidation is lost. We're going to be removing this terminal group here in order to donate some electrons. Okay, we're going to generate some G3P, the reduction step here, okay, because we're reducing this molecule. Okay, glucose and other organic compounds. G3P is our output here because 2 G3Ps are going to make glucose for us. But then we also have G3P. Notice that there's 6 of them here. 1 of them goes on to make glucose. That's why we need to do 2 times this whole reaction. Okay, 5 of them are going to go on because remember there's 3 here. And if we're doing 3 times 2 is 6. So 1 plus 5 is 6. This will make more sense if you read the textbook, I promise, I promise. Okay, we have 5 of these G3Ps that are actually going to go into the regeneration step to go make more RUBPs. Because if we don't have any RUBP, then there's nothing for our carbon dioxide to come in and attach to with the help of Rubisco, right? So if we don't have this molecule, then this whole thing's not going to happen. So only 1 of the, overall carbons that we put in, the 6 carbons that we put in, 1 of the things that we get out of the 6 total products actually gets to go on and become glucose. That's why we got to do 2 turns of it, okay? Again, this is exactly word for word in your textbook, written out very well with little blue numbers that indicate all the steps. There's little paragraphs that tell you about it all. Please go back and read this. Please. It's complicated. You just heard me explain just like a once over of this. There's even more detail than that. Please, please, please read it, okay? Okay, phase 2 is going to be reduction. So we talked about phase 1, carbon fixation. So we're putting our carbon dioxide with the help of Rubisco onto RUBP. Okay, carbon fixation, beautiful, okay? So cool. Phase 2, reduction. Involves the reduction of and phosphorylation of 3 phosphoglycerate. Again, so this is a phosphate in position 3 on a glycerate molecule. Oops. 3 phosphoglycerate, position 3, 3 carbons, okay, cool. Okay, to create G3P. 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 Notice that there's a couple things that happen in between, okay? Next, we have the phase 3, which is regeneration. So like I said, it involves the rearrangement of G3P to regenerate the initial carbon dioxide receptor or acceptor, which is the RUBP. Because remember, the very first thing that happens is that carbon dioxide comes in and it pairs up with RUBP because Rubisco helps to attach them together. Okay, that's creating our first product here, which is through carbon fixation. So we need to regenerate that RUBP. So we're going to use a lot of the G3P we just created to go on and create more RUBP for the next carbon dioxides that are coming in. Okay. This is talking about like the evolution of the overall, like why this process is happening the way that it does. So evolution of alternative mechanisms of carbon fixation in hot and arid climates. So other types of plants, you know, like cacti and like hot arid climates are going to do this whole process like pretty similarly, but like with minor differences. So adaptation to dehydration is a problem for land plants, sometimes requiring trade-offs while with other metabolic processes, especially photosynthesis. On hot, dry days, plants close their stomata. Remember that stomata are the little pores in the under part of the leaves that are used for gas exchange to allow the carbon dioxide in and the oxygen out. That's what the stomata are, which is going to conserve water because it also limits photosynthesis, right? If you close up those pores during a hot day, then those plants are going to hang on to more water. It's not going to be transpiring off the top of the plants. But if you are closing those stomata, then there's no carbon dioxide that's going into the stomata either, which means no carbon dioxide means you're not going to make any glucose. So there's no photosynthesis happening. So the closing of the stomata reduces access to carbon dioxide, I just said that, and causes a buildup of oxygen, which can be very bad for the plant, actually. These conditions favor an apparently wasteful process called photorespiration. Okay, so most plants, C3 plants are normal plants that don't have any special mechanism. It's just straight photosynthesis. Okay, initial fixation of carbon dioxide via Rubisco forms a 3-carbon compound called 3-phosphoglycerate. We just looked at that a few times. Again, pictures are going to be very helpful. In photorespiration, Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle, producing a 2-carbon compound. So photorespiration decreases photosynthetic output by consuming a lot more ATP, oxygen, and organic fuel and releasing carbon dioxide without producing any ATP or any sugar. But it's releasing carbon dioxide, which it's not able to get any other way because the stomata are closed in these plants that are doing photorespiration. So photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more carbon dioxide. So photorespiration limits damaging products of light reactions that build up in the absence of the carbon cycle. So again, photorespiration is when you are going to add oxygen instead of carbon dioxide in the Calvin cycle in order to produce a 2-carbon compound. Okay, it's also going to be releasing carbon dioxide here without the production of any ATP or sugar because you've used a lot of ATP and other organic fuels. Okay, but if you create any carbon dioxide, then you're still able to do a little bit of the carbon cycle in order to try to put enough carbons together to create some sugar. So this is just a mechanism to make sure that there's still something happening in order to get more energy. Kind of like in your body when you have like lactic acid, when you're like working out, that lactic acid burn is anaerobic cellular respiration. But if you didn't do that, you wouldn't have enough ATP to continue doing what you're doing and you'd probably die. So it's just kind of like a crappy way to get the job done. Like worst comes to worst, this is what we got to do. Sorry about it, but at least we're going to get the job done. It's going to be quick and sloppy and it's not going to feel nice. That's essentially what's happening here. Okay, we already talked about that. So C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into a 4-carbon compound. So this is different because photorespiration usually does a 2-carbon compound. This is incorporating carbon dioxide into a 4-carbon compound. Okay, so an enzyme in the mesophyll cells, remember the mesophyll is the tissue that's in the middle of the leaves, has a high affinity for carbon dioxide and can fix carbon even when carbon dioxide concentrations are very, very low. So even if there's only a little bit of like residual carbon dioxide because the stomata are closed, it can still be fixed. Okay, these 4-carbon compounds are exported to bundle sheath cells where they release carbon dioxide that is then used in the Calvin cycle. Okay, so this is showing you the overall like representation of the process. So you have a C4 plant and you have a CAM plant. So you have the mesophyll cells and you have our bundle sheath cells. So like I said, you're going to be actually like sending in a little bit of carbon dioxide with organic acids here. You're going to be releasing carbon dioxide in order to go through the Calvin cycle to produce sugar. Okay, it's spatial separation in the steps because you have two different types of cells. Here you have like it's all happening in the same area. It's temporal separations of the steps. Temporal is talking about like times of the day. We have night and day. So we have carbon dioxide that's going to be entering, mixing with organic acids, and in order to put more carbon dioxide into the Calvin cycle to create sugar. This happens during the day here, and then here this is happening at night. So you'd have those stomata open at night when temperatures are typically a little bit cooler, and then you'd close them during the daytime. So you're just getting this carbon dioxide from other chemical reactions that are happening without further input of carbon dioxide from the atmosphere. Okay, so canned plants, which is these I just talked about. Okay, so some plants, including succulents, are canned plants. They fix carbon a little bit differently. So canned plants open up their stomata at night, like I said, when it's a little bit cooler, incorporating carbon dioxide into organic acids. The stomata close during the daytime because it's very hot and they'll lose too much water and the plant will die that way. And carbon dioxide is released from organic acids and then used in the Calvin cycle. So that's basically what I was just saying, written out for you here. And there's the visual representation of it. Again, read it, go back and look at the picture. I promise the pictures really do help you understand this overall process. Again, spatial difference here because we have two different types of cells. We actually the steps occurring in different cells. And here we have it all occurring within the same cells, but it's temporal separation because of the time of day. Okay, so the importance of photosynthesis, a review. Okay, so the energy that's entering the chloroplast is sunlight. It gets stored as chemical energy in organic compounds. Okay, so we have the sunlight that's coming in. It's solar energy. The point of photosynthesis is to turn that solar energy into chemical energy. So sugar is made in the chloroplast. It supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Organic molecules, we're talking about like macromolecules and things here. You know, carbohydrates. We're making some carbohydrates right now in the process of photosynthesis. So plants store excess sugar as starch. Remember that like humans store the extra sugar as glycogen. Okay, that's like kind of like precursors to like fat and stuff like that. But glycogen is just a whole bunch of glucose repeating, and that's what starch is as well. It's just a little bit different linkages. So plants store excess sugar as starch in the chloroplast and in structures such as the roots, tubers, seeds, and fruits. In addition to food production, photosynthesis releases oxygen in our atmosphere, which is really important because that's what we need, right? It's not important for the plant, but it's very important for us. Okay, so this is the overall process that we just talked about. So they actually added in photosystem 2, the electron transport chain, photosystem 1, electron transport chain here. Okay, so we talked about the light reactions and how important it is to create these products that are then going to go into the Calvin cycle here to create that G3P, which is going to be released to store energy or to be utilized as when two of them come together to make glucose. Okay, that's going to be, it says sucrose exported. It's glucose that's exported. Okay, and then that RUBP is the thing that we are going to attach the carbon dioxide to as it's coming into the Calvin cycle. Again, right here in the picture, there's a little enzyme. It's called rubisco. That is the most abundant enzyme probably on the planet because it's in every plant. We need it, right? And there's a lot of plants. Cool deal. So that's the overall process of photosynthesis. Oh my gosh, did we do it? We did it, guys. We finished it. Photosynthesis. Please, I'm going to smash this and like, please don't have a seizure looking at all 1.5 billion of these slides. Okay, read this now, read this, make sure that those things make sense. Then go back through and look at all the pictures that I put in of the different steps. Okay, that's like the same picture. This, make sure that you can explain this. What is happening in each of these steps? This one, make sure that you go through and you read it. What is happening in these steps? Why are these things important? Where are these things going next? Why is this happening? Okay, then you need to make sure that you go through Calvin cycle is going to be the next thing. We just talked about the light. Okay, beautiful, cool. Okay, here we go. Calvin cycle. Read this. Look at the pictures. Make sure that this makes sense. Pause the video, flip this, make sure it all makes sense. Okay, I promise I put all these pictures here for a reason. Just like your textbook company, it prints all of these colored pictures for a lot of extra money for a reason. Okay, thanks for bearing with me. I know that this was a lot. I promise you that the study guides and the textbooks will help. Thank you. Have a great day.